So today we're going to talk about translation. And what we're covering right now is globally termed gene expression. DNA to RNA, RNA to protein. And gene expression is together transcription and translation. Transcription is making what? Making the mRNA from DNA. And we know the ins and outs of that, what proteins are involved, right? RNA polymerase 2, or RNA we just say polymerase, right? Transcription factors that help to do what? What's the role of transcription factors? Promoters.
They help to start the RNA.
They help to place it, right? They help to place where that RNA is going to go, right? Transcription factors, promoters. What's important about a promoter? What did we talk about in terms of what area of the TATA box, all right? TA, TAA, right? And that's sort of is important in placing where that RNA polymerase is so that you get the the right transcript produced. Okay? Now, you get a pre-mRNA made from RNA polymerase. That includes what? What is a pre-mRNA? That first transcript that's made includes what? Describe for me that mRNA that is produced by RNA polymerase. Single-stranded, it's RNA, okay. Is it ready to leave the nucleus, yes or no? What does it need? So processing. It needs a poly A tail. What else? A 5' cap. What else? Splicing. Right? Splicing involves what? Spliceosomes. And what is splicing going to do? Takes out the non-coding regions, which are called introns, and splices together the exons. Okay? Before we move on to translation, what happens if, say, we have an exon, an exon, an exon, exon 1, 2, and 3, and what happens if exon 1 is spliced to exon 3, or exon 1 is spliced to exon 2, and no exon 3? What is this called? Alternative splicing. All right? It's alternative splicing. What does it do? It changes the eventual protein. Okay? That's important in evolution, and in function of proteins themselves. Okay? And splicing is performed by spliceosomes, which are a mixture of RNA and proteins. Correct? All right, that catches us up to where we need to be. Now that mRNA is ready to leave the nucleus. All right? It leaves the nucleus, and what's its next step is going to be translation. Right? Translation. So, translation is an RNA-directed synthesis of a polypeptide. RNA includes those 3-letter words, right? The codons, which will eventually code for what? Right? Or the amino acids which will make up that protein. All right? So our genetic information flows from DNA to RNA, eventually to proteins. Okay? So translation is taking the code that's in RNA and arranging it, decoding it, translating it into the right sequence of amino acids for a right functional protein. Okay? To do this, what do we have to do in order to get the right amino acids in order? You have to be able to do what? I sent you a secret code, World War I. I sent you a secret code. What do you need to do to understand that message? You have to decipher the code. All right? So we know the codes are in 3-letter words, AUG, CGA, whatever it might be. All right? And we use that codon table to decipher code. So that AUG actually means a methionine comes in. All right? There's an intermediate between there. there's a step, there's a compound that actually works to decipher the code, and that's transfer RNA. All right? So that codon, that is that three-letter bit or word that's in RNA, is deciphered and and binds, is translated by the anticodon that is on tRNA. So if the codon for A is AUG on the mRNA, what is it going to be on the tRNA? What's the anticodon going to be? So here's AUG on the RNA. What's it going to be on the tRNA? UAC, right? So this is, why is it UAC? Complementary base pairing. We're still going by Chargraph's rules of complementary base pairing, right? So A to U, all right, and C to G. tRNA is made up of, has this area of the codon. So this is what tRNA looks like. All right, I'm going to minimize that. Give me this. I can't separate the two right now. So tRNA looks something like this. It is a molecule of RNA that looks something like that. Okay? So it's RNA. What do you know about RNA? Describe the characteristics of RNA, what it might be. Single-stranded. Does that look like a single-stranded molecule? No. It has three-dimensional structure. And the three-dimensional structure comes from complementary base pairing between parts of the single strand. Okay? So it actually looks like this. You have some base pairing that occurs. All right? This area here is where you get the anticodon. All right? I'm sorry, it was UAC that we were doing. Okay? That's the anticodon. And then this area up here is where you get an attachment to the amino acid. Right? So you have the attachment to the amino acid. Okay? So that what's going to happen is this tRNA, okay, is going to match up its anticodon to the codon and bring in the right amino acid, which then will be linked to the next amino acid, right, in the chain. And we'll go through sort of the process of that. All right? But the structure of tRNA is important in its function. Right? Structure-function relationship we've carried on throughout the semester and from last semester. All right? Structure and function related. Questions on tRNA? All right? So, working on translation, we're going to produce what? The end result of the translation is what, Nick?
Protein. Polypeptide.
Polypeptide. Why is it a polypeptide and not a protein?
Protein is made up of multiple polypeptides.
Some proteins are made up of multiple polypeptides, right? And that's that functionality distinction between the two. Okay, so here's the basic process, right? What carries out protein synthesis in the cell? Alexa, what is that? A ribosome. All right, so this is a ribosome. And ribosomes have two parts to it, and we'll go over the subunits in a minute. All right, so you have a ribosome binding to an mRNA, recognizing the codon, bringing in the anticodon, the right tRNA, right? What you see in blue here, and in purple you have the growing polypeptide chain. Simple, right? Not so fast, my friend. Okay? We know that there's a codon for every amino acid, right? Yes? AUC, methionine. Right? CCC is something else. CGU is something else. Okay? So for every amino acid, how many anticodons must there be? At least one, right? And at least one. We know there are 20 distinct amino acids. And how many possible combinations are there between a 3 base pair code, 64, all right? But, so at least every amino acid has one, or at least one specific anticodon, codon, okay? So each tRNA can translate a particular mRNA codon into a given amino acid. There are 3 codons that don't code for an amino acid, correct? Keep that in mind. Remember that. So every tRNA that has an anticodon can come in and bind to a codon, brings in an amino acid. But there are 3 that don't work, all right? And those are called what? Terminators, stop codons. All right, we'll come back to that in a little bit. All right? So the tRNA contains your amino acid at one end, right? And then on the other end, the anticodon, your complementary base pairing, which is for recognition, translating the code in mRNA. All right? Basics about tRNA itself, it's about 80 nucleotides long. It can base pair with themselves. That's what gives it its shape and structure, right? Flattened into one plane, like I drew it, it looks like a clover leaf, okay? But in 3 dimensions, it's sort of L-shaped, and you'll see what it looks like in a second. Here is your graphical depictions. Your clover leaf structure, right? Anticodon. You have the anticodon, right? Base pairing between the actual strands itself, and here's your amino acid attachment, right? Structure, function, it's what it's designed for. That's your 2-dimensional structure. Here's your 3-dimensional structure, and you see it's sort of L-shaped with the amino acyl attachment at the 3' end. Okay, and your anticodon sort of in the middle. All right, and then the symbol used in this book is, as you can see here. All right, pretty straightforward. All right, questions on tRNA. All right, what does the T stand for? Transfer. All right, because what's it going to do? Transfer amino acids to the right. All right, so the anticodon-codon relationship is important in accuracy, in bringing in the correct amino acid in the correct sequence at the right time so that you have a functional correct protein. You end up with the wrong protein, you end up with the wrong function, wrong amino acid, wrong function, wrong protein, doesn't do its job, right? So, first, it is important that the correct amino acid be linked to the right tRNA, right? So that you have, say, your clover leaf, right? And this codon, say, is AUC. That actually means that the right amino acid is attached here. You get the wrong attachment, you get the wrong protein, or the wrong amino acid put in the wrong spot because of the attachment between the tRNA and the amino acid wasn't correct, right? So there has to be some identification that occurs, an association that occurs between the right amino acid and the right tRNA, all right? And this is done by the enzyme amino acyl tRNA synthetase. So amino acyl meaning what? What does that sound like to you? Amino acid, tRNA, transfer RNA. This is the enzyme that's going to actually link amino acid to the transfer RNA. All right? And so this enzyme will recognize the amino acid and the tRNA and produce that bond. Okay? And then the second and probably the more important recognition is the codon-anticodon relationship. So that the right codon is recognized and the amino acid is placed in the right sequence. Okay? There is some flexibility that occurs. That third codon, and we know this from looking at the codons that are repeated. Right? If we went back to the chart, you can go back in your slides, go back to the chart, and you see that leucine has 6 different codons for it. right? And a group of those codons, a four group is like CGA, CGU, CGG, CGC, right? So in that third position, it doesn't really matter what amino acid's there, right? For leucine, it could be CGG. It doesn't really matter because regardless of which third one is there, leucine's going to be put there, right? So there is some flexibility in that third position, all right? So this allows some tRNAs to bind to more than one codon. You understand that? All right, so that, no, Abby? No? All right, so in that third position, okay, so you have, so here's your codon, say CGC, right? What's the anticodon going to be? GCG, okay? And that would be the correct base pairing. This codon codes for, let's say, I don't know if it's actually right or not, is leucine, okay? But what are other codon possibilities that could be for leucine? It could be, say, CGA, CGC, CGG, or CGU, all right, for instance, okay? So which tRNAs can fit in there? GCG, GCU, GCC, or GCA, right? So this third position then sort of becomes irrelevant because regardless of whatever tRNA goes in there, what's going to happen? What amino acid is going to be placed there? Leucine. So it doesn't change the overall sequence of amino acids, all right? And that third position allows some flexibility. Okay? Other questions on that? Better, Abby? You're welcome. Okay, so here's sort of how the amino acyl synthetase does its job. All right, so you have binding, here's the enzyme, it binds to the amino acid, and that changes its shape. So an amino acid, of recognition of what amino acid is there, allows for, changes the shape of the enzyme, and allows for the correct tRNA to bind and to allow. It's like lock and key fit, right? So that the tRNA that doesn't fit for tyrosine if it doesn't fit for tyrosine, it's not the right tRNA, it's not going to fit in that position once tyrosine is bound. All right? So the right amino acid stimulates the right tRNA to bind to the enzyme. Then the linkage is produced. All right? From there, the amino acyl tRNA is released, and this is an ATP-dependent reaction. Meaning what? It uses energy. All right? So, recognition involves linking the amino acid to the right tRNA and then a codon-anticodon relationship. Any other questions about that part of translation, that player in translation? All right. The next big thing is the ribosomes. So the interaction is great. So tRNAs are going to interact with the mRNA, complementary base pairing, etc. Great. Well, how do we make sure it just doesn't all happen at once? Or it doesn't start at the middle and then tRNAs just come in all together? This is an ordered process. And this all happens at the ribosome. All right? So first off, ribosomes facilitate coupling. They actually allow the tRNAs to find the anticodon, get them spaced correctly, and then you end up with the formation of proteins. Okay? Ribosomes are composed of two subunits. All right? They're mainly large proteins, and then with some RNA associated with it, ribosomal RNA. Okay? regardless whether it's prokaryotes or eukaryotes, the subunits, the large and small subunit of a ribosome usually are separated apart if they're not bound to an mRNA. If they're bound to an mRNA, they come together and you end up producing proteins. All right? So they only join, the subunits only come together when they're going to produce proteins. All right? This is important in how you start translation. Right? Because where do we want to start translation? With a start codon. Why have a start codon if you're not going to start at the right spot? Right? If you were going to start at the middle, what's the point of having a start codon? Or if it was just random, what's the point? There's a specific reason why we have a start codon. All right? At a certain point. Here's the structure of your ribosome. All right? You have a small subunit, a large subunit. All right? Two different parts, and they're usually separate. When they come together, they form these three sites. All right? Three spaces, if you will. All right? An E for exit site, a P for polypeptide site, and A for acceptor. All right? So you have the exit site. What do you think is going to happen at the exit site? What's going to exit? The tRNA is going to exit. All right? You have a peptidyl tRNA binding site. What do you think is going to happen there? What does peptidyl sound like? Peptide bond. That's where formation is going to occur. That's where your peptide is going to grow from. The acceptor site, or what's also called the amino acyl tRNA binding site. What's going to happen there? This is where new tRNAs come in. All right? And you'll see that this is a sort of a stepwise process. Right? Where here's your exit site, and you'll see that there are always, once translation starts, there are always an amino acid in the peptidyl site. Right? Either before exit or before another one comes in. There's always an amino acid, a tRNA linked to an amino acid at the peptidyl site. Otherwise, you get separation and end of translation. All right? This is your ribosomal structure. Right? Large subunit, small subunit, etc. All right, the P, the A, and the E sites. All right, polypeptide chain, the acceptor site where your linked tRNAs are going to come in, into the ribosome. All right, and then your exit site where they're going to leave once protein has been produced. Questions on ribosomal structure? So, the basic process. What do we have to do to start translation? We need a start codon. What do we have to do in order for translation to begin? What has to bind to that RNA? tRNA and a? How about what's going to produce ribosome? All right. So we have to sort of build the complex. This might sound familiar. We have to build the complex, let it go, and we have to tell it when it's going to end. What does that sound familiar to? Transcription. All right. Transcription occurred in three distinct steps: initiation, elongation, termination. Same thing occurs in translation. All right. Initiation. We build the complex. We get it started. Elongation. It does its job along, and then we have to tell it where it's going to end. Okay. Initiation, elongation, and termination. All three of these steps require certain protein factors. All right? There's certain proteins that are involved in getting everything set, keeping it going, and then ending it. All right? The initiation stage brings together your mRNA, your beginner tRNA, and then the two subunits of the ribosome. All right? What do we know about the 5' end of an mRNA? What's on the 5' end of a mRNA that has left the nucleus? The 5' cap, the guanine cap. Okay? There's a methyl, yes, there's a guanine methyl group on the 5' end. Okay? That 5' end tells you this is the 5, this is where we're going to start, and the poly A tail tells you where you're going to end. It gives sort of a directional part to the mRNA, right? Distinguishes one end from the other. Yes? Sort of. Yeah, you agree with that statement? All right. So, the 5' cap and a special tRNA actually stimulate the small subunit of the ribosome to bind to that area. All right? So we're going to start at the beginning of the mRNA because we have a signal that says this is the beginning. The 5' cap, this end is the beginning as opposed to the 3' end or somewhere in the middle. All right? Once the small subunit binds, that stimulates the large subunit to bind. Sort of so that you have small subunit here. Here's your mRNA, right? And now the large subunit binds sort of on top of it. And the mRNA is now clamped in between the small and large subunit, so it stays there. Right? This is sort of your translation complex. All right? The small subunit moves until it finds that start codon. That's when the large subunit binds. So now what have we done? What has the ribosome done? We've assembled a complex where along the mRNA? At the start codon. We've now find the right starting point. Okay? So here's what it looks like. Here's your start codon. So small subunit sort of places itself in the right position so that that start codon, AUG, is in the P site. That brings in the right methionine, large subunit binds, and now you have initiated translation. Okay? And there are other protein factors that help this to occur that you don't need to know about until you reach advanced molecular biology courses. Questions on initiating translation? All right. The start codon, it establishes reading frame. What do we know about reading frame? It's important. And thanks, Emily. It's important. Woo! Amanda, what do we know about reading frame? Since you're so awake this morning.
It's important. Going off of what Emily
Okay,
said.
why?
No idea.
Come on. What's going to happen if we're not in the right reading frame, Abby?
If you're not in the right frame, then you synthesize different proteins.
All right, you're going to synthesize different proteins. Why?
So 3 pairs.
Because the codons are going to be off. You're going to have the wrong codons. Right, Cody? You said that. Dina said that. Well, Dina doesn't say it out loud, so I can't. And she says, it's a methyl group, right? I don't know. I can't hear you. You didn't say it. All right? So, we position the small subunit along the start codon so we get the right reading frame. The methionine comes in. That stimulates the large ribosomal subunit to bind. Now the initiation complex is produced. All right? And then so initiation factors, and this is a very broad term. All right? It's very vague. No, I'm not going to tell you what they are. All right? There's initiation factors that now produce this complex that sort of complete it. You complete me. All right? Now everything's set up, elongation occurs. All right? Elongation in translation is a stepwise process. Okay? In order for elongation to occur, at this point in initiation, what do we have? Small subunit, mRNA, lined up the right way. We have a tRNA in the P site. Large subunit is bound. What do we need now? Now we're going to synthesize a protein. So we have the code. What's right? So we need the amino acids to come. We need the next amino acid to come in. Correct? So where does the amino acid come into? It comes into the A site. All right. So right now we have sort of, let's say this is a large subunit, small subunit. Right. Here's your P site, and this is occupied by a tRNA. Right. Here's the A site. And here's the E site. Okay? The A site's empty. So a tRNA is going to come in. All right? Once that occurs, then a reaction is going to occur and it's going to produce the peptide bond between the P site and the A site. Right? The amino acids there. And then this whole thing is going to move over. The ribosome is going to move over. So that now the tRNA that was in the A site moves over to the P site. The tRNA that was in the P site moves over to the E site. What does the tRNA that's in the E site not have anymore? An amino acid. So then it doesn't have an amino acid. It is released from the ribosome, goes off, and goes find its other amino acid. Okay? So it sort of slides down. All right? And you'll see this in this lovely graphic you see here. Okay? So here we are at the beginning. P site is bound. You notice every one of these steps, there is an amino acid in the P site. Okay? That's where the growing polypeptide chain is going to come from. Okay? Beginning 1, codon recognition. Here, tRNA comes in based upon what codon, anticodon is there. All right, with its amino acid, binds to the A site. This is stimulated by hydrolyzing GTP to GDP. What does that sound like to you? What does that mean? Energy is utilized. All right, so this comes in. You get formation of a peptide bond. All right? Now, the ribosome moves over. Again, it translocates. Right? From moves down a step, a 3 base pair step. Now that P site, what was in the P site is now in the E site, and then it's released, and now it sets up for the next amino acid to come in. Questions on this? Emily?
What is G in GDP?
Guanine. So what is A in ATP? Adenosine, right? So this is basically a nucleotide that they use to break down energy, G for guanine, right? Similar in structure and function to ATP, but not as utilized in the cell. Okay? This is elongation. This occurs as long as there is a codon, anticodon recognition. There is a amino acid or a tRNA that has a growing polypeptide with it in the P site, right? This termination. Termination occurs when we reach a stop codon. That's your signal, right? So a stop codon does not code for what? An amino acid. It doesn't have a recognition that occurs. All right? So when a stop codon reaches the A site, instead of a tRNA coming in, there's a protein that comes in. what we call a release factor. All right? That release factor causes the addition of water, right, instead of an amino acid. So water comes in, it doesn't produce a peptide bond. It produces a different type of bond, which then stimulates a change in shape and a dissociation of all those things. Ribosomes separate, mRNA separates, tRNAs are released. Now that's the end of translation. That protein can go off and fold and do its job as necessary. All right? So like this. Here's, instead of stop codon, UAG, UAA, or UGA. All right? It doesn't code for an amino acid, so a tRNA doesn't bind there. Instead, this protein binds there. The release factor then is the addition of water. All right? What is not happening here?
It's not moving to the piece.
There's no translocation. Right? It can't move over. So it doesn't transfer the peptide to the next tRNA. All right? So it doesn't move over, causes hydrolysis, separates the tRNA from the growing polypeptide, and eventually get dissociation from everything. Right? And then again, this is energy dependent. Right? GTP is broken down. Questions on termination? All right? Questions on the process of translation? Okay. On Monday, we will go over, so we've sort of we'll go over some of the ideas of what happens to a protein from here, and then we will also go over regulation of gene expression. Right, we'll start, how is it that these transcripts are regulated so that a gene isn't produced when it's not supposed to be produced? Okay? Questions?